So this is just introduction to microbiology. Now for this very short session, we have objectives, we have learning goals. Okay, first is to describe the historical development of the science of microbiology and its relationship to medicine and other areas of biology. Then very briefly, we will review the nature of microorganisms. And lastly, we will discuss the scope, relevance, and future of modern microbiology. So, what is microbiology? Nag-stock up na tayo.
Study of microorganisms.
Di ko siya gusto mag-klase. Okay, so this is the study of organisms and agents too small to be seen clearly by the unaided eye. Kaya siya micro. So basically, this is the study of microorganisms. So we need a microscope to appreciate these organisms. Now, how did the world discover microbes? And how did they associate microbes or microorganisms with disease? Okay, so we start with our great-great-great-grandfathers. So as early as 98 to 55 BC, we have this Roman philosopher, Lucretius. Together with another physician, they suggested that disease was caused by invisible living creatures. So, dili makita sa eyes, pero maka-cause of disease. Okay, so, gi-argue nila ang mga previous belief na magkasakit ka because you are cursed, di ba? Ano mo na, witchcraft before. And then, he was supported with this physician, Girolamo Fracastoro. Actually, he was the first to establish or first to propose germ theory. Okay, so he suggested that diseases were caused by invisible seeds. Seeds pang iyang description. That could be transmitted from one person to another. Okay, so that starts the germ theory. Next, so di man daw to makita ang katong maka-cause of disease. Eventually, later, we have Anthony van Leeuwenhoek. He was the first person to observe and describe microorganisms accurately because he was also a microscopist. So he made a microscope, crude one. Okay, so based on his findings using his microscope, there are animal cues. So he sent letters of his discoveries to the Royal Society of London. And here it is clear that from his descriptions that he both saw bacteria and protozoa. Okay, so this was his microscope. Be thankful. Okay na itong microscope karon, no? Did you use your microscope during histology?
Yes.
Very good. Because this time, you have to use also microscope in microbiology as well as in pathology. And this can magnify 300 times the actual size of those microbes that he saw. So these are the drawings of what he discovered. Okay, so you see the round objects there. They could be horcocci, okay, and the bigger ones are the protozoa. Okay, so they also believe in this theory, spontaneous generation. So this is a hypothetical process by which living organisms develop from non-living matter. Grabe, no? Non-living tapos na maka-produce ka of living organism. So this theory is how they explain the origin of life. So dili ka tungkuan. Sige, itong origin of life na to, itong monkey, ikantas monkey. Oo, lahi po ilaha, spontaneous generation. So according to this theory, for example, ngano man nila nigi, ingon from, you can produce a living organism from non-living because, for example, you will put a cheese and bread sa rack in one corner, dark corner, and then later on, makaproduce daw siyang mice. So, di ba, bread non-living. Tapos eventually na ay mice. So, dito nila gi-refer ang kaniyang theory. Okay, so do you believe in that? Very funny siya, no? Because this was long time ago. Wala pa man, di pa makaayon to this rudimentary pang science. Okay, so there was a conflict of this theory. So that view was challenged by this Italian physician, Francesco Redi. So he carried out series of experiments using decaying meat and its ability to produce maggots spontaneously. So meat again, and then it can produce maggots. So he used 3 containers. One was uncovered, the second one was covered with paper, and then the third one is covered by gauze. And then what happens next? In the presence of that meat, flies are attracted. Of course, they are more attracted to the open one because they can enter, and then take a look at the image there. What happens? So after the fly go to the meat, maggots are produced. Okay. However, those bottles or containers which are covered, okay, there are no flies inside. There are no maggots, except for the one that is covered with gauze. On top of the gauze, there are flies there, and eventually it produces maggots. Why? Because the gauze has tiny holes. So ma-attract hapon sila because they can smell it. Okay, so that's why they will stay here on top, but they cannot enter. So what's the difference from the spontaneous theory previously believed by the people, and from this experiment of Redi? So may inyong ma-conclude. I-agree ba siya sa sa spontaneous generation? No. So mo to siya gi-challenge ni Redi ang katong spontaneous generation by doing this experiment. Okay, was not able to generate maggots except when they are in contact with the meat. And it's the fly that produce the maggots and not the meat. So according to him, belief unconfirmed by experiment is vain. So kailangan ng facts. So dili ta magpataka og marites, no, mao po na atong lesson diri. Okay, more than put akong cursor. Anyway, next, in 1748, there was this English priest, John Needham. He reported the results of his experiments on spontaneous generation. So he boiled meat and broth, and then cover it with flask, and then eventually many of the flasks became cloudy and it contained microorganisms. So this one, it contradicts Redi's claim. Kasi si Redi, kung covered lang siya, wala yung spontaneous generation. Pero sa kanya, he boiled it, then after which he covered the flask. Pero naging cloudy pa rin yung broth niya. So still, meron pa rin. It contained microorganisms. So he thought organic matter contained a vital force that could confer the properties of life on non-living matter. Then few years later, Italian priest and naturalist Lazarus Spallanzani, he improved on Needham's experimental design by first sealing the glass flask that contained water and seeds, and If the sealed flask were placed in boiling water for 3/4 of an hour, no growth took place as long as the flask remained sealed. So the difference from katong kay Needham, gipa-boil sa iya, then after that, dito pa niya gi-cover. Kang Lazaro, he covered it and then boiled it. Okay, so he proposed that air carried germs to the culture medium, but also commented that the external air might be required for growth of animals already in the medium. So that's why prior to heating, gi-cover na niya because naay kanang naay roll ang air para daw mag bring in sa microorganisms. The supporters of spontaneous generation maintain that heating the air in sealed flasks destroyed its ability to support life. So actually, because of these experiments, this is also now they're also establishing the sterilization method. Okay, so we have other supporters of biogenesis. Okay, according to Theodore Schwann, he allowed air to enter a flask containing a sterile nutrient solution after the air had passed through a red hot tube. The flask remained sterile. While George Friedrich Schroeder and Theodor von Dosch, they allowed air to enter a flask of heat-sterilized medium after it had passed through sterile cotton wool. No growth occurred in the medium, even though the air had not been heated. And we also have Felix Fouché. He carried out experiments conclusively proving that microbial growth could occur without air contamination. So all of these arguments are from the previous experiment na na-roll ang air. Okay, so these are the supporters of biogenesis. That's Theodore. This one is Theodore von Tosh, and this one is Felix Fouché. So what is this biogenesis? This is opposite to spontaneous generation. Okay, biogenesis is when living things can only be produced by another living thing and not by a non-living thing. So i-contradict niya si spontaneous generation. And those are the pro-biogenesis supporters and their examples. So who finally end the spontaneous generation controversy? Who do you think? No idea? Regarding sa mga sterilization? Yes, it's Louis Pasteur. Okay, so what did he do? So he made an experiment. He filtered air through cotton and found that objects resembling plant spores had been trapped. So if a piece of the cotton was placed in a sterile medium after the air had been filtered through it, microbial growth appeared. So he made this experiment. Take a look at the flask. Naala gid yung design, or there's a purpose for that. Okay, so he made this another experiment. He placed nutrient solutions in flask, heated their necks in a flame. Diba naala naman ka mag-sterilize prior to performing, for example, inoculating microbes into your medium. Okay, so i-heat muna natin siya. Then, drew them out in a variety of curves while keeping the ends of the necks open to the atmosphere. He then boiled the solutions for a few minutes and allowed them to cool. So what's the importance of this shape of the flask curve? Okay, so in this experiment, he noticed that no growth took place even though the contents of the flask were exposed to air. Di ba open man ang flask? But look at the shape. Then he pointed out that no growth occurred because dust and germs trapped on the walls of the curved necks. Mo na siyang importance why he used these curved necks. But if the necks were broken, growth commenced immediately. And because of this, he had also shown how to keep solutions sterile. Next, we have John Tyndall. He is an English physicist, and he also argued with the spontaneous generation together with Louis Pasteur. He demonstrated that dust did indeed carry germs, and that if dust was absent, broth remained sterile even if directly exposed to air. So in the air, there is dust, and dust carry germs. During the course of his studies, he provided evidence for the existence of exceptionally heat-resistant forms of bacteria. So eventually later, this heat-resistant forms of bacteria was discovered by Ferdinand Kohn as bacterial endospores. Okay, so what is the role of microorganisms in disease? Now, the importance of microorganisms in disease was not immediately obvious to people. So it took many years, many scientists to establish the connection between microorganisms and illness. If you can still remember itong Black Plague, diba naghanap tayo mga movies about that? So before, they say that you have this because you've seen, you're cursed, witchcraft. Okay, so later pa nila na establish na that is caused by yes, by a bacteria. So the recognition of these microorganisms will depend on the development of new techniques for their study. So how do we identify microorganisms in the lab? Naman dito mga mentex dali, no?
James. Mamona na.
So, mag-start ang gram stain. But, if you really want to identify, sa ganun na siya ng procedure, we do, sa nga stain. O, katulad dito nga table, o, sige sila, o, smile. Kabalo sila sa answer. Yeah, I can do gram stain, but you can just classify them into gram-negative bacilli or cocci. What else? And you can really identify the species. Culture, right? We do culture. Pero karon kay machines naman ta karon, no? I think sa inyong time, machine na. Punta na lang dito ang kuan culture battle sa machine. And then the manual method is more fun because you will really see the reactions. Anyway, okay. So how did we recognize the relationship of microorganisms and disease? Because of this one, germ theory of disease. So they support this theory, and many scientists developed this, and one of that again is Louis Pasteur. So kisa gani itong first nag-start, propose ang germ theory. Ito sa first few slide na ko.
Siguro, Girolamo.
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 Oh, basta kadto siya, no? So, siya ang nag-first propose, and then later, ipadayon siya ni Louis Pasteur.
Girolamo.
So, Pasteur proved that a microscopic plant caused the souring of milk, that is lactic acid fermentation. And then Pasteur was able to prove that living cells, which are actually the yeast, okay, were responsible for forming alcohol from sugar. So, we have to thank him for the alcohol, why you have alcohol now. And that contaminating microorganisms found in ordinary air could turn the fermentation sour. Next, he identified microorganisms responsible for both normal and abnormal fermentations. and found that through heating wine, beer, milk, or vinegar briefly, certain living organisms could be killed, thereby sterilizing. So, the word pasteurizing or pasteurization. Now, this leads to Pasteur's thought that if germs were the cause of fermentation, they could also be the cause of contagious disease. And so he began to develop this germ theory of disease. And aside from that, in developing the germ theory of disease, eventually he also worked on vaccinations. Okay, so as early as from the time that Pasteur is actively working on germ theory, inanapod ka, karaan na ang vaccination. Right? So, are you a believer of vaccines? Nagpa-vaccine mo ng COVID? Nakay nag-smile. Oo. Because at that time, dagang kay mga anti-vaccine, right? So, as a doctor, you know the truth that you have to educate the people. Dagang kay mga belief sila ba? Although, it will take time talaga to develop a vaccine, but they know already the principle. Again, the side effect, it will take time, years. Okay. So next we go to this another remarkable person, Joseph Lister. So may na-contribute ni Lister. He is the father of antiseptic surgery. Yes. So actually, first endeavor niya is on the prevention of wound infection. Surgeon man siya. So he was so impressed with Pasteur's studies on the involvement of microorganisms in fermentation. So he said, maybe I can also use that in sterilizing instruments. So what he did, after heat sterilization, he also used phenol. So phenol can kill bacteria. And this one, Listerine. Okay, but this is not from Joseph Lister. Naalay na kanang siya na-encourage from his work of kanang sterilization, antiseptic technique. So he was able to formulate this Listerine from Lister. Okay, and this was first used as an antiseptic for sa mga OR. Okay, then eventually, nahimo na siyang mouthwash. Okay, do you use this? Oh, you know na dako ang the history of Listerine.
Surgical antiseptic.
Then finally, we have Robert Koch. So the first direct demonstration of the role of bacteria in causing disease came from the study of this man in the anthrax disease. Okay, so he is a German physician. So to establish the relationship between Bacillus anthracis and anthrax, he published his findings. How? Through this experiment, first, he used mice. Okay, he injected healthy mice with material from the deceased animals, those animals with anthrax. And then after that, the mice became ill. So we have to assume that healthy mice got the anthrax. And after transferring anthrax by inoculation to a series of 20 mice, he incubated a piece of spleen. Wala pa man sila culture medium at that time. So a piece of spleen containing the anthrax bacillus in beef serum. Then from that growth, bacilli grew, reproduced, they produced spores. So when these bacilli were isolated, injected into the mice, again that mice develop anthrax. So his criteria for proving the cause causal relationship between microorganisms and a specific disease are known as Koch postulates. And this can be summarized as follows. First, the microorganism must be present in every case of the disease, but absent from healthy organisms. Second, the suspected microorganism must be isolated and grown in a pure culture. Next, the same disease must result when the isolated microorganism is inoculated into a healthy host. And then the same microorganism must be isolated again from the diseased host. So one of his experiments, actually, when he's a coach, he also used himself as the subject of his experiments. So I forgot already, I think nagkasakit na yata ni siya TB or because of this one. Pero bawal naman na siya karon, unethical na siya karon. Before, pwede pa mag-good. Wala pa sa ethics committee. Okay, so the germ theory revolutionized the practice of medicine and hygiene as it led to the development of antiseptics, disinfectants, sterilization, antibiotics, and immunization. Because of this also, natabangan ta during pandemic, right? So, diba nag-hoard man tataghan kay antiseptic, disinfectants. Before musulod sa balay, maligo. Have you tried that? Oh, food. Inagitan sa hospital, maligo sa bawas. So, at least, because of this, we were saved during that pandemic. Okay, so we're done already with the history. Let's proceed now to the brief review of microorganisms, the nature of microorganisms. But I will not discuss in detail because again, you will discuss this in the next topics when we go to bacteriology. Okay, so take a look at the origin of microorganisms. Very old na. They existed on Earth for about 3.5 billion years. Una pa sila sa atua. And, okay, the structure, we have two cell lines. We have the prokaryotes and the eukaryotes. Cellular na sila, and the acellular virus. The lifestyle of microorganisms, they are found everywhere. They exist everywhere. In air, soil, water. Okay, some microorganisms have close associations with other organisms. There are also that will cause harm. We have parasites.
Ang mga tao.
Okay, next, we go now to the scope, and relevance of microbiology. So, modern microbiology is a large discipline with many different specialties. So, dili lang kaning ina-discuss na to. Dili lang kaning subject actually. So, it has a great impact on fields such as medicine, agricultural and food sciences, ecology, genetics, biochemistry, molecular biology. So you can also pursue here the other specialties of microbiology. So example, public health microbiology, this is closely related to medical microbiology. What we have now, or what is your subject now. Okay. Here, it will try to control the spread of communicable diseases. They will also monitor community food establishments and water supplies in an attempt to keep them safe and free from infectious disease agents. So we have this activity in the laboratory, kaning monitor water supplies. So kani siya, lingaw ni siya kay you will get water supplies from the different areas here in school. You can also use outside. And then later you will know if the water they're using is contaminated or not. Then another specialty is the agricultural microbiology. This one is concerned with the impact of microorganisms on agriculture. So here, they will try to combat plant diseases that attack important food crops, work on methods to increase soil fertility and crop yields, and study the role of microorganisms living in the digestive tracts of ruminants, such as your cattle. And currently, there is a great interest in using bacterial and viral insect pathogens as substitutes for chemical pesticides. Another one is microbial ecology. So this study the contributions of microorganisms to um, the relationship between microorganisms and their living and non-living habitats. This one, the study of pollution effects on microorganisms is also important because of the impact of these organisms have on the environment. So, in bioremediation to reduce pollution effects. For industrial microbiology, here microorganisms are used to make products such as antibiotics, vaccines, steroids, alcohols, other solvents, vitamins, amino acids, and enzymes. Now we also have microbial physiology and biochemistry. Here, they will study the synthesis of antibiotics, toxins, microbial energy production. And the most recent one, microbial genetics and molecular biology. So they focus on the nature of genetic information and how it regulates the development and function of cells and organisms. So genetic information, gene mapping is not only done in human, but also in microbes. They're also concerned in producing new microbial strains that are more efficient in synthesizing useful products. So we have these laboratories producing microbial strains. Okay, so siguro isa na dito si COVID, no? Ingo nila nag-originate na sila sa laboratory in China. So it's not illegal. There are laboratories doing that. Genetic techniques are used to test substances for their ability to cause cancer. So for treatment. And the more recently, the field of genetic engineering. Engineered microorganisms are used to make hormones, antibiotics, vaccines, and other products. Okay, so what is the future of microbiology? So to mention some, okay, one, new infectious diseases are continually arising, and old diseases are once again becoming widespread and destructive. So what are these re-emerging diseases? Smallpox, nagbalik na ba si smallpox? Katong monkeypox? Oh, pero na-contain lang man to, no? AIDS. Dugay naman na si AIDS, pero re-emerging. Hemorrhagic fevers, one of that is dengue. Okay, so be careful because pangit ang dengue karon. And even TB, re-emerging siya. So microbiologists will have to respond to these threats, and many of them are still unknown. So sila ang mga lab rats. Then microbiologists must find ways to stop the spread of established infectious diseases. So even if TB, established naman na siya because of antibiotic resistance. Now, microorganisms are increasingly important in industry and environmental control. Okay, so here are the use of microorganisms in biotechnology, in genetic engineering, in bioremediation to control pollution. And lastly, microbial communities often live in biofilms, and these biofilms are of profound importance in both medicine and microbial ecology. Kaya siya, minsan dili ka mag-respond sa imuhang antibiotics because of biofilms. So they have to study how to penetrate these biofilms so that your antibiotics will become effective. And you will again encounter these biofilms in the other topics. Okay? So, do you have any questions? Happy sila, okay, tapos na po. Yes, very short lang din siya, and this is just an introduction to microbiology. So, actually my goal here is just to, to open your minds na microbiology has many specialties, not only medical microbiology. Okay? I will give you a PDF copy of this. I-learn. Sa I-learn ko na lang ilagay ha. PDF lang ha. Okay, before I will leave, gentlemen, it is the microbes who will have the last word. Okay, so do not, ay magbagot-bot mo, sige mo memorize in microbiology. Okay, because you know what? Ang payoff magyapon ng inyong effort sa sige memorize because during board exam, dalian ninyo ma-recall. So, based on experience, usually sa mga takers, and even sa akong personal experience, okay, because grabe ako memorize. So, during review for the board exam, less na lang na time akong gihatag sa microbiome because pabalik-balik naman ko din sila, o magoy mausab. Right? Di mausab ang pangalan, o naalay gamay, pero same right hapon. Unlike sa others, like, you really have to study a lot, the issue, everything. Okay? So, good morning everyone.
What's the smallest sa universe? Dito sa sabay ka pa pag-lunch ka ba? So yun ang kasi mga pharma.
Gusto ko lagi si Doc.
Alam mo, dito ang sugo.
Si Doc Dean. Kasi official group with Dean diri ah.
Official. May official word.
Kani. Amo ni na grupo. 165 man.
Tutubi.
Aha man.
Napay 1 hour. Napay 1 hour. Tension of hour.
Taon na lang tadaan.
Kani.
Introduction to immuno. Wala na yung orientation.
Ay, dapat ko siya nga siya mag-send of schedule.
Wala man late na ko nakasulod.
Morning go. Ano? Introduction window. Wala na yung class sa morning.
Hindi, dapat si Doc Pandalan.
So yung mamaya, night class mamaya, move na ngayon. Wala na night class. So wala tayo 5 lang.
Yes, yes, Lord, yeah.
Ano, ituro ni Doc Bandalan yung immune.